lecture. It will be last lecture in 2023. And we have a great young energetic speaker today from Australia, Erin Grant. Let me introduce Erin. So Erin Grant completed her master's and PhD qualifications at the University of Melbourne, Australia, in quantum biosensing. Erin's work was focused on developing a clinically relevant test of ferritin-bound iron using the nitrogen-vacancy defect in diamond. Currently, Erin is undertaking postdoctoral training at the Commonwealth Scientific and Industrial Research Organization as a part of the newly formed Quantum Biotechnology Group. Erin worked on nanoscale NMR using fluorescent nanodiamonds. Erin, please, your lecture can be started.
Thank you. I'll just share my screen. All right, thank you for the introduction, Olga. Before I begin my lecture today, I'd just like to acknowledge that I'm speaking today on the lands of the Wurundjeri people, who are the traditional custodians of the area on which I'm speaking. And I pay my respects to their elders, past, present, and emerging. So I'm going to be covering a recent publication that I worked on over the last few years, which was developing a new technique for measuring T1 relaxation from fluorescent nanodiamonds while they're dispersed in solution. So I'll be covering the methodology and the protocol that we use to do these measurements, and also the motivation for why we developed this technique in the first place. Okay. So as you're probably all aware, there are quite a number of uses for fluorescent nanodiamonds that have been demonstrated. So from fluorescence tracking, pH, free radical temperature, and paramagnetic ion sensing. Primarily or commonly, they're looked at from the perspective of biological sensing applications because of their small size. We can do nanoscale level measurements. And they're not cytotoxic, so they can be taken up by cells. And so we see a number of examples of that here. What's interesting is, although we can use nanodiamonds for doing kind of basically all of the protocols that you would do using a single crystal diamond containing NV defects, a lot of the biological applications, so if we consider pH, free radical, and paramagnetic ion sensing, the majority of those are done using T1 relaxometry. So all of my work in my PhD and master's was focused on sort of looking at paramagnetic targets and therefore doing T1 relaxation. And so we will be considering that in this talk. So in case you just happen to not be super familiar with T1 relaxometry, I'll give kind of a brief overview and how we implement it in nanodiamonds. So the NB defect has this electronic structure with triplet ground and excited states. We can polarize the center into the ground zero state using off-resonant green laser light. And then we can get an understanding of the spin state by looking at the intensity of the red fluorescence that it emits. T1 relaxometry just looks at the longitudinal relaxation from this ground zero state. And in the absence of any kind of external target, we will get an idea of sort of the intrinsic processes in the diamond, mostly thermalization processes due to phonon interactions. And so when we do a control measurement, we just turn the laser on, let the system interact for some length of time, tau, on the x-axis here, and then probe the spin state again. And we build up a decay curve that looks something like this. So this is defined by the T1 time, which is 1 on E of the maximum value. But in all of our results, we present this as a relaxation rate, so just 1 on T1. If we have a nearby target that we're looking at sensing, this will produce magnetic fluctuations that are on resonance or overlapping with the transitions in the ground state, and this induces relaxation, and so we see a faster decay rate with a target present. And so we're looking at changes, basically. So as I said, many biological targets produce these magnetic fluctuations, and typically they have a very wide frequency spectrum. So that's represented here by the blue curve. So several orders of magnitude in frequency. But the filter function of the NV defect when we're considering T1 relaxometry is very narrow, and so we're able to probe this frequency spectrum. So when we consider just a single NV defect, if we look at the relaxation curve, we will get a single exponential function. But when we're looking at nanodiamonds, primarily, or all the work that we do, we're looking at nanodiamonds with an ensemble of NV defects within them. And we also are looking at an ensemble of nanodiamonds. So we will have a very large number of relaxation rates being contributed to the overall signal. And each one of these, due to the fact that each NV defect is in its own sort of unique environment, will have a slightly different relaxation rate associated with it. And so that means we can't fit the data using a single exponential. We need to have slightly different fitting functions. And there are sort of two main approaches to doing this in the literature. So the first is using a double exponential of the form shown here. And this works well in situations where we have sort of two major timescales, so a short and a long relaxation rate, and the contributing decay rates are sort of clustered around these two timescales. The second approach is to use a stretched exponential function, which is of the form here, where we have extra power on the exponent. And so this is better suited to cases where you have a really wide distribution of, or sort of a continuous distribution of contributing relaxation rates. And so we looked at three different sizes of nanodiamonds, so 50, 100, and 140 nanometers in this paper. And so this is from data that I'll present later on, but we fitted that data using both of these fit functions and found that when you look at either the long relaxation rate in the double exponential, which is sort of typical, or just this relaxation rate here, not considering a target, just the nanodiamonds by themselves, you can see that the associated fitting errors are smaller for the stretched exponential case. And so we think this better accounts for the sort of distribution that you have in these particles when you're considering sort of a large number of FNDs. So all the data I present from now on will be fitted with a stretched exponential. And I thought I'd just flag that because there's been some work recently looking at sort of which approaches are better. So I think it's something that we need to consider when we're doing T1 relaxometry. So as we all probably know, although there's a large opportunity to be using FNDs in a wide range of settings, we haven't really seen the transition from sort of the research community, quantum sensing groups into sort of biological groups or just general uptake. And that's due to the fact that there are a few challenges that are remaining that we need to address. And there are two major ones. So firstly, if we look at a given sample of nanodiamonds that all sort of have nominally the same size, when you look at them under a TEM, you see that there are actually, each one is quite unique, and you get quite a large distribution in diameter and aspect ratio. And so this is some nice work from RMIT that shows that quite well. And then the other sort of main issue is that because of the high surface area to volume ratio of nanodiamonds compared to a single crystal diamond, the effect of surface spin species is important. And so when we look at the quantum properties of nanodiamonds, we see that they sort of on average have about a factor of 2 worse quantum properties than a single crystal. So these are some challenges that we need to address moving forward. And it really means that we need sort of good characterization techniques that allow us to look at an ensemble of nanodiamonds so that we can understand, you know, if we do some kind of intervention, some kind of fabrication method, we can assess how that affects the sample on average, and with high throughput, basically. So just go over some of the pros and cons of sort of standard ways of measuring nanodiamonds and how that kind of led us into the development of our in-solution technique. So I just want to flag that we don't see this new method as sort of superseding any of these techniques. It should be complementary and offer like just slightly different information. So typically nanodiamonds are looked at on a single particle level. And obviously, you know, if you want to use them in biological context, we do want to look at them at a single particle level. And so this is usually done with a confocal microscope. So it gives you, you know, good precision, allowing you to do particle tracking and offers high spatial resolution. And in terms of characterization, you can correlate spin measurements with other measurements like TEM, for example. However, the cons of this technique is that it means that it's difficult to understand ensemble behavior. You know, you can only measure maybe hundreds of particles and it takes quite a long time. So it is a difficult technique to use to characterize new FND samples. And as well as that, if you're looking at sensing, because it's a 2D format, it makes it a bit difficult to measure dispersed targets because from one perspective, you know, you're only really interacting with your sample on one side of the particle. And it just offers challenges in terms of like how you actually implement things. The second kind of standard technique is using wide field microscopy or wide field measurements. And in this technique, you can measure the ensemble of FNDs. And that means that you can characterize sort of the average behavior of nanodiamond samples. And if you attach a camera as your detection method, you can also get spatial resolution. However, I have done some wide field imaging of nanodiamonds, and I've found that it's quite difficult to create a uniformly dense sample. You're sort of reliant on just how the nanodiamonds naturally settle onto a cover slip. And if you're looking at imaging, you need to also apply microwaves to the sample so that you can cancel out background noise. And so when you combine sort of the fact that you have nanodiamonds that are sort of settled in random clumps, they might not be in the best place for your microwave resonator. Again, because it's in a 2D setting, it's sort of a bit difficult to implement measurements for dispersed targets, and you might need sort of microfluidic devices. In comparison to confocal microscopy, I would say that you get medium throughput, but yeah, there are still some sort of practical challenges. So that leads us to sort of why we wanted to move towards a slightly different methodology. So all the work that I did in my PhD was looking at measuring a protein, and we wanted to understand the average behavior of this protein. So we're looking at it in solution. And I did my measurements actually on single crystal diamond, but the same kind of concepts could be said for like nanodiamonds on a cover slip. Basically, you know, the interaction between the diamond and the protein kind of occurs over long periods of time. So we were trying to think of ways to increase the interaction time. And then looking at sort of characterization techniques, we were also thinking about how to measure a large number of nanodiamonds with high throughput. So this sort of led us to be inspired by immunoassays. So there are types of immunoassays where you have suspended particles. So these would be like polystyrene balls, and they're covered in antibodies. And then these interact with your dispersed target quickly, and you can either measure them while they're dispersed, or you can kind of pull them out of solution. And so we thought you could do something similar with nanodiamonds while they're dispersed. And this would allow you to measure an ensemble, have fast interaction times, and hopefully use a pretty simple apparatus to do it, with the potential for miniaturization and being able to sort of give it to non-experts to use. So this is the apparatus that we created. So it is pretty simple, it just consists of a laser, an AOM for modulating the laser. And then we use just kind of a standard qubit to hold the nanodiamonds and the target analyte. we measure the fluorescence at 90 degrees and with a single photon detector at the moment. So you can see this is an image of what the actual cuvette and holder looks like. If we have a filter in front, you can see that the nanodiamonds produce quite a lot of fluorescence in this narrow band here where the laser comes in. So this is the sensing or the protocol that we use to do T1 measurements. So it's an all optical measurement where we just pulse the laser on and off. And then what we're measuring is the signal at the beginning of the laser pulse and dividing by a reference at the end of the laser pulse. And then we repeat this whole sequence as many times as necessary. So because this was something that we hadn't measured before, we wanted to do quite a lot of validation to make sure that this protocol was measuring T1 properly. So we went through a number of sort of steps to look at how it's performing. So the first thing that we did was identify the optimum laser pulse length for this protocol and to get good polarization across the entire measurement length. So we're using an illumination intensity per nanodiamond that's similar to wide field. And if you look at just applying the laser for 100 microseconds, you can see that once we get to about 70 or 80 microseconds in length, we get a saturation of the fluorescence intensity. And then if we look at different laser pulse lengths using our protocol and just consider the intensity in the reference window, so this is when the system should be repolarized for the next measurement, we would expect that if we're applying this laser for a long enough period of time, you would see the same intensity in each reference window. And so this is the data here. And you can see that once we get to between 60 and 100 microseconds, we do get a leveling out of the intensity as we would expect, but there is still a small modulation. So we decided that 80 microseconds appears to be sort of the lowest modulation that we get. So we settled on that as our laser pulse length. I will say that we did this for different sizes and different nanodiamonds from different origins. And you do sort of need to calibrate this for different particles. So we wanted to study that modulation that you still see to see if we can account for those changes. So we expect this could be due to charge state changes or laser intensity fluctuations. So we know from this work from Anya J.H.'s group from a few years ago now that we get charge state changes during the laser time and in the dark as well. So we need a way to account for these. So what we did was use a strong magnet to investigate spin-independent effects. So similar to what Angelo Buffoni's group in Italy did, where if you apply a strong magnet, you can get spin mixing in the ground state. And this means that you essentially can't polarize the system, and you can look at changes to the intensity that are spin independent. So we were then looking at sort of charge state changes. And what we did was look at what happens in the signal and the reference window across a T1 measurement. And so we did that with just nanodiamonds by themselves, which are this green data, and then with a target and a light there as well. And you can see the modulation is pretty small in percentage, but it is the same in the signal and the reference window. So that means if we divide signal by reference, we should be able to cancel out these effects, which is good because obviously we're using an all optical method here, so we need to be able to account for those changes. So the next part of the optimization method was to look at what the width of the signal window should be. So if we have a wider signal window, we're collecting more photons per sweep. And so that means that we should reduce optical noise per sweep. But that also means that we're reducing the contrast of the measurement. And so we want to optimize or find the sort of sweet spot between those two contributing factors. So if we look at a given T1 curve for signal window width, so changing this width here. As you can see, for the narrowest width, we have data that has a high contrast, which you can see plotted here, but there is more noise on the measurement, and that changes as we go to a wider width. So this is the contrast of the measurement, and this is the fitted relaxation rate and the associated fit error, which you can see plateaus and the error decreases. So this is the fit error in that value. So we found that the fit error plateaus after about 10 microseconds. So we chose that as the width of the signal window because it maximizes the contrast, basically. Okay, so the final thing that we wanted to check before doing sensing measurements with this technique is what concentration we can use of the nanodiamonds. So you can see if we look at concentrations between 1 and 300 micrograms per mil, the clarity of the solution changes quite a lot. So at 1 microgram per mil, it's pretty transparent, whereas at 300, it's basically opaque. And so we think that this would have an effect on the measurements, and indeed it does. So if we look at the contrast of the data, that sort of abruptly decreases after 25 micrograms per mil. And this translates basically to data that is a lot noisier and you can't fit properly. So, we get values of the relaxation rate that kind of don't make sense. So for these particles, we're kind of limited to 25 micrograms per mil or less. Actually, we chose 10 because that sort of seems to give you, relatively fast measurements, but the associated noise is lower. But this is dependent on the size of the particles that you're looking at as well, because scattering increases with particle size. So if you're looking at smaller nanodiamonds, you could probably operate at higher concentrations. However, if we're thinking about the sort of concentration of the target analytes we might be sensing, we don't really want high concentrations anyway. Okay, so once we had sorted out the sensing protocol and we were happy with sort of all the optimizations that we'd done, we did an example characterization study. So how we might use this, in this case, to choose which size nanodiamonds are currently best to measure in this setup. So sort of ideally, a smaller size particle should be better because more of the NV defects will be close to the surface of the nanodiamond and therefore able to interact strongly with the environment. But there are sort of challenges with smaller particles at the moment in terms of charge state stability and getting enough NV defects in them. So we wanted to sort of compare 3 different sizes. So we did actually look at 30 nanometer particles as well, but they basically, you couldn't really measure them in this setup. And so we compared just the 50, 100, and 140 nanometers. These were from Adamas, these particles. And so we're looking just at the intrinsic properties here. So if we look at the relaxation rate, a lower relaxation rate is optimal here. So the 100 nanometer particles are performing well, and obviously a higher contrast as well. So we chose 100 nanometer particles to work with from this point, although the 140 were also very good. I'll just flag over here, this is the power on this function, and because it's quite similar across these particles, it sort of indicates that the distribution of contributing relaxation rates is kind of similar across all of the samples. So the next thing that we did was perform a sensing experiment with these particles. We used gadabutryl monohydrate as the target. So this is a good molecule to use sort of to characterize samples and understand sensitivity because it has a single gadolinium ion at the center of the molecule with a spin of 7.5, so it has a very strong signal. But it's also neutrally charged, so it shouldn't interact with the diamond surface, which could kind of make it a bit difficult to sort of compare different samples. So to do the sensing measurements, we first take a control measurement in an appropriate buffer In this case, it's just milliQ water because we don't need a special buffer here. But if you were going to do measurements on something that required a specific pH or specific salt levels, using the FNDs in that buffer is important for your control measurement. And then we would do the control measurement and get a value for the relaxation rate of the sample and also the power P. Then add the target and do a second measurement. And in this case, we use this function to fit the measurement with the target. And we have used the value of the intrinsic relaxation rate and the stretch power that we got from the control. And then we just extract the effect of the target. So you can see we made a relaxation versus concentration curve to sort of understand the behavior with concentration. The intrinsic or control measurement that we did is the same across all of the samples as you would expect. And then when we add different concentrations of the gamma-butyl, we see the sort of shape again that we would expect. We wanted to make sure that the gamma-butyl wasn't interacting with the particles and potentially causing charge state changes or anything like that. So we looked at the fluorescence spectrum of the nanodiamonds by themselves and then with the target, and we didn't see any significant change there. And then also looking at the effect of the target for a given concentration over several hours, there's not really any trend in the data. So again, we don't really think that there's interaction over those timescales. So some more recent work that I've been doing is actually now using this in solution technique to go and characterize new samples that we've been creating. So I've been working with Mina, who's a chemist at Bio21, to investigate whether we can improve the inherent properties of the nanodiamonds by addressing the surface noise sources. So we know that changing the surface functional groups has an effect on what the noise sources are. And Mina had experience doing silica functionalization, which is kind of a common technique in a lot of nanoparticle studies. But in the context of nanodiamonds, it's mainly been used to sort of make them a more uniform shape and smooth out their surface. But it is changing, obviously, the functional groups on the particles. And so we were interested in whether this has an effect on the T1 time. So Mina used the Stoiber process to create particles with varying thicknesses of silica shells. And you can see 3 of these here in these TEM images. So we have one that doesn't have a complete shell, another with a 7 nanometer shell, and then this one has a 12 nanometer shell. And we compared the intrinsic relaxation rate to bare particles. So you can see here we again want a particle with a low relaxation rate. And once we have a continuous shell, we see a decrease in the relaxation rate. So the silica does have an effect. And very impressively, the 12 nanometer shell has a relaxation rate that is about the same as a typical HPHT single crystal diamond. So this is really indicating that the FNDs have internal structure that is able to house long relaxation times. And if we address the surface appropriately, we can change the surface noise sources and reduce them. But we have obviously increased an offset between the nanodiamonds and their environment with this silica shell. And so we wanted to see how that affects their sensitivity. So T1 sensitivity scales as 1 on R to the 6th, sorry. So when you introduce an offset, that has a big effect on the sensitivity. And you can see that over here, if we look at those same particles, the 12 nanometer shell has the lowest sensitivity. So that really does impact the way that we can use them for T1 sensing. So I did a bit of modeling to understand whether there is a point at which you can win if you introduce silica. So if we assume that the introduction of any thickness of silica will reduce the surface noise density, I then did a Monte Carlo simulation using this recent work from RMIT, where they looked at sort of the distribution of different sizes and found that they're all sort of ellipsoidal in shape. So I used this information and generated ellipsoidal particles, and then looked at the change in surface noise density that we sort of see between the bare and the silica-coated particles. And this is the results of that simulation for different sizes of nanodiamonds. And for all the different sizes I looked at, if we had a 1 nanometer shell, we would expect to be more sensitive than the bare particles. And so this is very interesting and sort of gives us a direction to move forward in terms of if we wanted to further optimize the Stoiber process. But in practice, it could be quite difficult to actually physically do that. But I think this study is very interesting from the point of view that surface functional groups are very important for the intrinsic properties of the ND defects. I'll also flag that there's recent work from Peter Mora's group in Chicago, who did a very similar thing and found that this application of silica also improves the decoherence rate of the nanodiamonds. So whatever the changes are to the surface functional groups, it's able to affect a very broad frequency range in noise. So I'll just finish now by just sort of thinking about what we hope to do with this technique and how we hope to improve it. So at the moment, each measurement to get a good enough photon statistics takes about 50 minutes, which is obviously not like ideal where we want it to be. So we're going to improve the throughput by modifying the detection part of the apparatus. So at the moment, because we're using a single photon detector, that gave us really good time resolution for validating the protocol, but it means that we're not capturing most of the fluorescence that is actually emitted by the nanodiamonds. So we're pretty close to kind of settling on a new methodology using a photodiode and increasing the size of the incident beam. So that should be able to hopefully drastically increase the throughput. We also want to move towards miniaturizing the system so that we are able to kind of do more collaboration and sort of disseminate this technique to other labs. And so, yeah, we're very keen to hear about, how we can use this method to look at a lot of different questions and sort of help with characterization and things like that. So thank you for listening. I'm happy to take any questions.
Erin, thank you very much for your interesting talk. Yeah, it's very exciting direction. If you have questions, please either ask directly, unmute yourself and ask a question, or type them in the chat.
Yeah, I have a couple of questions. Yeah, thank you for that. Yeah, great talk. So 2 questions. Why do you detect at 90 degrees? Because wouldn't you get a lot of scattered light versus detecting at, say, like 100, like back detection or like backscatter detection? Because I think you'd get a lot of scattered light as opposed to fluorescence emission at 90 degrees as compared to like 180 degrees detection. And the second question is kind of unrelated to the first. So, on the slide where you showed the gata-butyl, I believe, why at 10 to the -4 does the target give, if I'm interpreting this correct, like the blue, I'm looking at the top right graph.
Yeah.
So would I interpret the R, like INT, is basically without the gadolinium present? Or
Yeah.
I'm just trying to understand why in the 10 to the -4 it goes
Below.
like it's actually lower, but on the other ones it's higher. Yeah.
Yeah. So I think that's basically just, that's below the detection limit. So I would say we're unable to detect really at that concentration. So it's just within the noise. And so it just happened to have like come out that way.
Okay. But just in this case, your error bars don't, it's, yeah, I agree. It's probably below the detection limit, but the error bars of the individual measurements don't overlap, right? In these, between those two data points. Okay. Yeah.
Yeah. Yeah. Yeah.
Okay. Yeah. I think, yeah, it's just the detection limit. Yeah. Okay.
Yeah. So well, the other question, that's an interesting point. We haven't actually tried doing that. I think partially it's just sort of the ease of setting up the optics is kind of easier to do it in that direction. And then also you have to think about how you remove the incoming laser from what you're detecting. Because obviously they'll be in the same beam path, so you need, you know, like dichroics and things like that. And we are using quite at the moment a very high laser intensity. So there's sort of just like practical considerations on making sure that you're staying within the specs of like the intensity that you can put through your optical elements and things like that. But that is something that I think we could think about.
Cool. Thank you.
Hi, great talk. I was wondering, how many nanodiamonds do you think you're actually probing, like based on the size of the beam and the qubits in one of these 50-minute measurements? And do they kind of drift in and out of the excitation volume?
Yeah, so at the moment we're measuring at the concentrations that we're looking at now. With a rough calculation, it's around 2,500 to 3,000. Which ideally we would like to get more. So that's why we're going towards, well, partially why we're going towards a larger beam size. And if you look at sort of what you would expect in terms of drift, we don't expect them to move very much in the beam just because of their size. Like if you do the calculations, yeah, they shouldn't move too much.
And then these are like at high fluorescence, so they're multiple thousand. There's like 5 times that nano NV centers if there's that many nanodiamonds.
Yeah.
Okay.
Yeah.
Cool.
I have a question on how do you account for fluorescent background, which can contribute to the signal? So if you have a sample with fluorescent background, for example, biological sample.
Oh, okay, yes.
So say not in these situations that you described, but how these T1 measurements can be.
Yeah, that is actually an issue. It's quite difficult because if it's a pure, like for example, my measurements with protein, if you look at like a serum sample, for example, which has a lot of protein in it, that's very fluorescent. So you basically can't do the measurements. But if you're looking at like a sort of a typical biological buffer, like Heapy's buffer or MES buffer, something like that, that's fine. You can do measurements in that. So yeah, I think if you have like lots of proteins in your in sample that you're looking at, that is very fluorescent. So yeah, it is an issue. And you kind of need to think about how to purify the sample down into something that's like mostly salts and things like that.
And I have another question. So you just mentioned buffers or salts, And there was recent paper on increasing Q1 relaxation time in the presence of high salts. It's paper by Butcher. So did you observe increase in Q1 similar to that paper? It was done on diamond crystals with shallow NVs, and it was 2 times higher Q1 relaxation time at, I believe maybe it was 10 millimolar maybe of sodium chloride.
Yeah,
And they
don't.
did a simulation, and there is a model why it happens. So how you can account for that potentially?
Yeah, all of the measurements that we've done, I'm just trying to remember if that's similar concentrate. I feel like the biological measurements may be at lower concentration of salt than that. I have to check. But I haven't observed an increase in T1 with a buffer, basically. If anything, I would say you probably get a bit of a decrease, a small one. But yeah, I haven't observed that. So when that paper came out, we did discuss looking at higher concentrations of salt and seeing whether we could observe something like that. But yeah, we haven't done any measurements on it. So yeah, it would be interesting.
Do we have more questions from the audience?
Maybe just one follow-up question on the scatter-butyral. So at these concentrations, the particles themselves maintain like good colloidal stability at these concentrations of scatter-butyral?
Yeah. We don't really observe any change. The concentration of the nanodiamonds is quite low. So 10 micrograms per mil is much lower than sort of like the standard concentration that you just get nanodiamonds at. So I think possibly that has something to do with it. Yeah, most of the nanodiamonds that we've used have pretty good colloidal stability.
Alex, maybe another question, really, since we have the slide, I was looking at your dependence of relaxation rate versus concentration. From here, it's very difficult to determine what would be the sensitivity limit. I would expect that if you assume just that, as you said, dipolar interaction and dipolar type of noise, and you have a bulk solution, so basically rate should be simply proportional to concentration. Linearly proportional. You have log plot, but then you have a range up to, let's say 10 millimolar, where its data essentially flat. So there is not much of dependence there, and then one data point that shows that actually at 100 millimolar you do see significant measurable change. So I don't know what is if you're looking from just perspective of analytical type of measurements, people like to actually to determine what would be sensitivity limit, what minimal concentration you can detect.
Yeah, I would say at the moment, these measurements, there is a bit too much noise associated with them, so it does make it difficult to determine things like that.
You obviously know you always have noise, but what current noise, you can determine actually what sensitivity limit is, just simply I don't see much, enough data to
well, I guess the point of these measurements was not really to determine things like that. And I agree, like if we did want to use it as a standard curve to try and actually understand things like sensitivity limit and actually using it to measure unknown samples and things like that, we would do more replicates, more measurements, and sort of look into what the dependence is that we would expect. So I can't remember off the top of my head, but you do expect, depending on the concentration of the particles, although I think it's different because it's in a molecule. if like these were free ions, you would have two linear ranges where sort of the contributions, the frequency spectrum kind of changes depending on what concentration you're looking at.
But if you assume basically, if you assume, you know, gadolinium, you know, 3+, you know, electronic relaxation in solution is fairly actually fast. You know, you of course you may allude to that you also have effects on protons, you know, relaxivity in this case. So I don't know, yeah, because
yeah, I think that curve itself would need more measurements. Okay, so for example, this measurement here is a bit odd. So I think we would need to go back and look at that measurement again. It's quite, I think these measurements, because this system is quite sensitive, it's sensitive to like how the samples were prepared. And I have to say our lab is not the cleanest place. So we're a bit worried, you know, that potentially if we were going to create a proper standard curve, we would need to do it in a situation that was a bit cleaner, more replicates, and just be very careful about how we prepared the target solution. So yeah, I would not use this curve to look at unknown samples, if that makes sense. It was more to just show that we can do these measurements and get an understanding of, you know, does the relationship look basically how we would expect it?
Okay.
Can I ask another thing? Just so maybe kind of just, well, but does it matter? Do you have an idea of like what the concentration of the target has to be for your, like, in kind of relative to the concentration of the nanodiamonds for you to making sure you're getting a good like interaction and signal from it?
Yeah, so we did consider this. So in the case of non-interacting target and nanodiamonds, as long as your signal is above background noise, it shouldn't matter, like, yeah, it shouldn't matter too much. So you should be able to resolve the influence of the target given enough replication of your protocol. Basically, you know, you should be able to get to the point where you're eventually resolving the effect. But ideally, so if we consider the case of like sensing proteins, where we want to engineer an interaction between the target protein and the nanodiamond, so we would create some kind of specific binding interaction, then obviously we want to be able to capture all of the target on the surface of each nanodiamond and to use up all of the surface area that's available. So in that situation, you really do want to think about, okay, what's the available surface area of the particle versus the expected concentration of the analyte, so that you can really match sort of one-to-one, I guess. So yeah, there's kind of a few things to consider when you're thinking about things like that.
Erin, I have another question. So you are pursuing these ensemble T1 measurements. Do you think ensemble T2 measurements are possible in principle?
So you do, in those situations, you need to use microwave driving, and the particles are obviously dispersed and moving around, so you're not going to get good coupling between the microwaves and the NV defects. So I think it would be pretty difficult. Like, you'd have to do some pretty fancy pulse schemes to be able to ensure that you're actually interacting properly. So in my position currently, we're kind of thinking about how you would do that. for like the idea of doing like NMR and things like that, but sort of coming at it naively, it would be pretty difficult to integrate microwaves into this system. Also, from the perspective of the geometry of a qubit is very large, so you need very high microwave power to be able to address things within the qubit. So yeah, we're really at the moment just thinking about it from the perspective of T1. Yeah.
And what's about ensemble T2 on powder samples, on nanodiamond powder?
I think you have the same kind of issues because all of the orientations of your NBs will be random, like in a similar way to the qubit. So that's why pretty much all, to my knowledge, all T2 measurements are done on individual particles. Yeah.
Yeah, but it's definitely a big challenge, but also if overcome, very useful technique.
Yeah, yes, it would be.
So quantum physicists will be kept busy for next years, right?
Yes, yeah.
Okay, do we have more questions? You were answering for quite a long 20 minutes or more. So thank you so much for your very insightful talk. It's always great to hear new developments, and I wish you luck in 2024 in your new position, new job, and hope to see you at some time at some conferences.
Yeah. All right. Thank you very much for the invitation. It's been lovely.